Hey folks, I'm going to try to keep tonight's session short and sweet. I only have a couple little topics that I want to cover. This is the leftovers of evolution. So this is session 2, and I have a little presentation for you. So give me 1 second, I'll get that set up. Oops. Screen share, application. Yep. All right, let me present that. And we're going to shrink that down so I can see you. All right, so if you have any questions, don't be afraid to put something in the chat box. And let me know. So just real quick, we're going to just run through a couple things. So, this is actually the end. Hang on 1 second. Oh, there we go. Come on, baby. There we go. So, part 2 of evolution, we're just going to kind of finish up all the loose ends that we haven't done. We did some Darwinian evolution last time, and we talked about some other little topics. So I just wanted to kind of finish up. I could do an entire session on one of these topics, but I'm going to kind of hold off for now. So again, I'm Mrs. Carell from Kettle Run High School. I teach AP Bio and Bio. If you're not one of my students and you happen to stumble upon this, you can follow me on Twitter at Carell Science, and my YouTube channel should have whatever reviews I do. So last time we talked a little bit about divergent evolution, the idea that organisms evolve away from each other, they diverge, and that can be due to all kinds of reasons. But tonight, what I want to talk about a little bit is how species form, how we end up with different types of organisms from divergence. So speciation is defined as the formation of new species due to evolutionary change. So there's 2 little definitions for species. The more popular one right now, the one that science teachers teach typically, is the biological species concept. It's basically where if a group can interbreed and produce fertile offspring, then you deem that a species. It prevents things like hybrids from being considered species. It prevents things from organisms that might be genetically compatible but never run into each other from being the same species. It does cause us to redefine things like the dog because it can interbreed with a wolf. So meanwhile, there's what's also known as the phylogenetic species concept. This is probably more popular in the scientific world rather than the high school academic world. The phylogenetic species concept is basically accepting any small group of organisms that can interbreed with each other and share a common ancestor. They don't have to be as tightly knit as the biological species concept makes it. And this is going to work better for things like salamanders that are a little harder to tell apart. It may not be as obvious who's who. So the question is, how do new species develop? What is it that causes this divergence, that causes organisms to become so separate that they no longer interbreed back and they can no longer be considered the same species under the biological concept? So, and by the way, these are jumping spiders, which there are tons of, and each one has their own unique coloring and dance. And this is a beautiful photo that I did not make. I've got a link to it at the end. It is gorgeous. But yes, these are just some of the species that have been identified recently of jumping spider, all completely different. So one of the first things that we think of when we think of why species diverge and never come back is this concept known as reproductive isolation. The idea that individuals that maybe could have been the same thing can't be anymore because they can't reproduce. That can be either prezygotic, which means before they even mate or before they even are able to create a zygote, or it can be postzygotic. This is after they mate, they produce a zygote, but the zygote's going to not have a good success rate. So prezygotic barriers can include things like behavior, organisms that don't mate because they have improper mating behavior. They don't do the right dance. They don't have the right call. We were watching in one of my biology classes today a bunch of birds of paradise try to dance. And it's funny because the birds of paradise are not really that genetically different. It's just the idea that they've evolved to be so separate behaviorally and appearance-wise that they just don't even recognize each other as the same species anymore. And so we don't recognize them as the same species. Temporal is prezygotic, the idea that some organisms mate at different times than others. So even though maybe they could mate, they don't. And so that could be something like an organism that only mates in the day versus the night, or only mates in certain seasons, and they don't run into each other and they don't mate at the same time, so they don't end up mating. Also prezygotic would be just living in different locations. They never encounter each other. So even if they were reproductively sound together, they won't be because they don't run into each other. They don't have overlapping territories. Even if they wanted to mate, even if they had interest in each other, other prezygotic barriers include things like gametic issues. The egg and the sperm won't work together. Chromosomal problems in the egg and sperm that causes them not to resolve, or maybe they don't recognize each other cellularly. It could also be mechanical. These two organisms may be interested in each other, but the parts don't work. Their reproductive organs are incompatible. You can have certain animals where, what's supposed to be there is in the wrong place or is the wrong shape, and then they cannot actually physically mate. All of these would prevent a baby. On the other hand, postzygotic barriers are, yes, they can make a baby, but the baby doesn't work out. So reduced hybrid viability would be, okay, you had kids, but the kids don't really do very well. There's certain organisms where when they mate, like salamanders, their babies just don't do very well at all. Maybe your babies are okay, but your babies aren't okay. That's known as hybrid breakdown. So you see this in plants sometimes, that they'll breed for a generation or two, but by the third or fourth generation out, that plant is now failing. And it has to do with how their genetics line up. The last one, and the one people know the best postzygotically, is reduced hybrid fertility. And the most famous example we always mention is mules. This idea that you can make mules by breeding a horse and a donkey together, but mules can't make mules. So if you need more mules, you have to breed more horses and donkeys. And that's because the horse chromosomes and the donkey chromosomes, while they have basically the same genetic signatures, they don't have the right chromosome numbers. The horse actually has an extra that is a donkey chromosome split. And so when the egg and the sperm come together, you can produce offspring, but those offspring just have errors and it doesn't work out. So while that organism is fine, it can't make any more organisms because meiosis fails. So these are all after you have a baby, the baby doesn't perform well. And that's going to cause you to say these organisms are no longer the same species because they can't reproduce together. Some other reasons why species diverge, other reasons why organisms end up so far apart that they're not the same species anymore. There's what's known as allopatric speciation, and this is geographical isolation, the idea that these organisms no longer have contact with each other. Over in my little diagram over here that I borrowed, there's beetles, and the beetles for some reason got separated, it looks like by a river or something. Well, because they were separated for so long, they went on their own evolutionary pathways, and over time, even if you reintroduce them, they're no longer the same beetle. They will no longer recognize each other. Maybe they have other issues. It could be prezygotic or postzygotic, but the idea is they've been separated for so long that even if you reintroduce them, they're not the same thing anymore. Sympatric speciation. So watch the names here. Allo is apart. Sympatric, just like synchronization and synergy. Even though they're together in sympatric speciation, they diverge for some reason, even though they're overlapping. This is really common in plants and not in animals. In plants, you'll have an organism that ends up with some sort of different genetic signature, usually polyploidy. Instead of having maybe 2 sets of all its chromosomes, it ends up with 4 sets. Well, now it can't breed back to the organism that it started as. And so that organism thrives. It makes lots and lots and lots of offspring with the same genetic signature. And so you end up with a brand new species emerging within a parent species. So they are now 2 completely separate and distinct species, but they started in the same place. They didn't have to be isolated like these little beetles did. Another type of divergence that you'll see, another speciation that you'll see, is what's known as adaptive radiation. And this is the Galapagos finches that Darwin saw. This is African cichlids and South American cichlids, the fish. So this is where you have an original ancestral organism, in this case, a seed-eating ground finch, kind of started it all. It blew onto the islands. And then its offspring, who were slightly different, went off and took other niches. And it's not because they wanted to or needed to. It's because they were better suited to eat different food types and deal with different conditions. And so over time, you end up with all these different types of finches that radiate out from the original finch. And none of them recognize each other as the original finch or as each other. And so they create all these behavioral isolation techniques that they no longer interbreed. And now they're all officially separate species under this umbrella. It's really fascinating. So now that we understand why we have species, how do these species change under time or over time? And so remember, natural selection may act on an individual. You might be starving, or you might have the advantage. You might be better at getting food, or you might get eaten by a predator, or you may evade a predator. But it's not you that evolves, it's your population that evolves because of you. And so a population in biology is defined as any group of organisms of the same type, interbreeding individuals, a species, within a place. That's the difference between population and species. This population is locale-based. Okay? A gene pool describes the genes of that population. And so you'll hear people comment on gene pools and things like that. And it's just this idea of what genes are available in a population. If it's not in the gene pool, it's not going to show up unless there's spontaneous mutation. So what influences a gene pool? Well, gene pools are mostly influenced by things like natural selection. If a gene gives an individual an advantage or a disadvantage, but if they do get an advantage, maybe they pass that on and the frequency of that gene increases over time. Gene pools are also influenced by gene flow, which is when organisms move in and out of the population due to migration, and they bring genes with and take genes away. Genetic drift is a really interesting concept. These are usually very sudden events that change the gene pool drastically. The two big famous genetic drifts are what are known as bottleneck and founder's effect. Bottleneck effects is when some devastating event limits the original population down to a very tiny new group. So you can see in the picture why it's called bottleneck is literally you picture marbles in a bottle. And if you pour those marbles into a glass and the glass are the only ones that survive, the bottle's gone. Those surviving organisms go on to create a new population, which may no longer reflect the genetics of the original pool. And that's because, as you can see in this glass, instead of lots of blue marbles getting in like were in the original bottle, most of the marbles that made their way to the glass were red. And so our final population, our new population, is mostly red, not blue. And you will see this sometimes, especially when a species is very, very severely limited. If there's a big extinction event or something like that, a disease. Meanwhile, founder effect, this is kind of interesting. So the original population stays, that's not gone, but a subgroup of the population branches off and becomes a new population. Not far enough to be a new species yet, maybe eventually. But what's interesting is the founder population doesn't represent the original mother population here, just like that little glass on the bottleneck didn't represent the original bottle. And so here, that little founder population goes off, and it may be that the organisms that come from it have a very specific gene pool that you maybe don't even see all that often in the regular pool. So it's like a kiddie pool to the big pool. And if the kiddie pool doesn't represent the big pool, you get this interesting effect. In animals, one of the examples I love to give is wild horses that get isolated. So the feral horses that live off the coast of Virginia on the Chincoteague Island and Assateague and Chincoteague. Or, for example, I usually talk about the Keeger Mustangs. They look completely different than other wild horses because their founder genes are different. They represent a different color pattern, a different body build than a regular wild horse. It's kind of fascinating. All right, so what happens to a gene pool if nothing happens? Let's say that there's absolutely no drive for any differences to occur. Well, then you would expect that your gene frequencies would be the same forever. And this is a concept known as the Hardy-Weinberg principle, or the Hardy-Weinberg equilibrium. Now, this one always throws kids for a loop because in essence, when I teach it, I tell my students, it's not a real thing. The Hardy-Weinberg equilibrium basically doesn't exist in nature. What it is it's a tool to show us that evolution does work. So if you're going to have no evolutionary drive, so I said on here, to get Hardy-Weinberg equilibrium, you have to have no evolutionary drive. So no evolutionary drive means there's 5 assumptions. Your population has to be huge. I have a picture of a panda down here because 1, I hate pandas, and 2, they're a great example because there's just not enough pandas for this to work. The population has to be huge, and that's because you have to have a large, stable gene pool. You can't have any selective pressures. So there can't be any issues with food, space, mates, competition, anything, which good luck with that. Pretty much any organism will eventually outgrow what it's supposed to do. There can't be any mutations, which is physically impossible. Every time DNA is copied, there's always some sort of mutation. Most of them get caught, but some always sneak through. And especially in organisms like bacteria, where it's so quick, mutations are a thing. I mean, that's actually how bacteria evolve. So, and then finally, there can be no migration. So you can't have any members of the population being added or removed at any time. So again, good luck with that. And all mating must be random. There can be absolutely no reason why anyone picks their partner. And we've seen in evolution that is not the case. A lot of evolution is driven by females or males picking their mates based on particular characteristics. So any sexually reproducing organism, you're going to have a really hard time finding completely random matings. Really, what Hardy-Weinberg is doing is it's saying if there's no evolution, then nothing changes. It's such a weird statement. Now, there's a cute equation that some of you are probably familiar with, b plus q equals 1, or p squared plus 2p cubed plus q squared equals 1. We will do something else with that later. I don't feel like calculating it right now, but basically it's just to back this up. And assuming that a population doesn't meet these 5 requirements, because it won't, you will not see Hardy-Weinberg equilibrium, but you will see a predictable change in genetics depending on what changes are occurring in the environment or for that organism's pressures. All right, so the last thing I want to talk about real quickly, I'm not going to go too far into this because really it's a rabbit hole of awesomeness, but the last thing I want to talk about is this idea of cladograms and phylogenetic trees. And just to kind of review that these exist, there's a lot of overlap between cladograms and phylogenetic trees. You'll sometimes see them labeled as each other. People are not that picky about it. The big difference between a cladogram and a phylogenetic tree is that a cladogram is usually based on physical characteristics, and a phylogenetic tree is usually based on some sort of genetic comparison. So it could be DNA, it could be RNA, it could be rRNA, it could be proteins, amino acid differences, whatever it is. It's usually a little more technical than, hey, this thing has hair. Cladograms tend to be a little more primitive, but they don't have to be. There can be really complicated cladograms. Sometimes phylogenetic trees can involve dates. This one does. They can also involve other things. But either way, really quickly, when you're talking about a phylogenetic tree or a cladogram, so for cladograms, I like to teach the bus rule. You start at the bottom where the bus picks up all these animals. And so in this case, these are all, well, they're all chordates. They're not all vertebrates because the lancelet is not a vertebrate. It doesn't have a true spine. But anyway, so all these animals get on a bus together and you start chugging up the road and you hit the first stop. We get to vertebral column. Well, there's a problem. The lancelet doesn't have a vertebral column. So he has to get off the bus before he can move on. Okay. Meanwhile, everybody past that, you would infer, has a vertebral column. So we're going to keep going. Next stop, jaws. Well, sorry, lamprey, you're a jawless fish, so you have to jump off the bus. So then we're going to keep going on the bus. Oh, 4 walking legs. Well, sorry, tuna, you have no legs. And so then we keep going up. And then in this case, what's setting this mammal apart right here is the presence of hair. But because this mammal got all the way to the end, that tells you this mammal has hair, an amniotic egg, 4 walking legs, jaws, and a vertebral column. And it's a chordate. and it's an animal, and it's a eukaryote. So the farther you get up the chain, the more unique aspects you have that things lower on the chain do not. Now, sometimes we make mistakes with cladograms. We just don't know. A lot of times they're just hypotheses and we're just kind of trying to guess. And as we learn more about what sets organisms apart, we have to correct that. So like, let's say you didn't realize that, turtles also have an amniotic egg, and you try to put the turtle down here. Well, then when we find out the turtle has amniotic eggs, oops, we move it back up along the line, and it makes it a little closer to mammals than we might have thought. Over here in the phylogenetic tree, you start at the root, which would be the ancestor of all of these organisms. And then as you hit a node, that's a divergence. So this organism branched off. This is yeast. So yeast branched off. And then all the other organisms, you're saying they're more closely related to each other than they are to yeast. Okay. Then the next branch, C. elegans, that's a nematode worm. Okay. Well, it's not as closely related to all these other things as, other things, but it's more closely related to these organisms than yeast is. And so here's their common ancestor, here's their common ancestor, here's their common ancestor, here's their common ancestor. And in this particular phylogenetic tree, they actually tried to give you dates to tell you basically when the divergence occurred. They don't all do that. Sometimes they'll have numbers to indicate the number of different amino acids or the number of different base pairs or whatever's been tested. This one came from a paper where they were trying to compare proteins in different organisms from these categories. So, but anyway, it's always unique. Every question's a little different. You might run into a question where they basically give you a table of differences between different organisms, and your job is to categorize them, to put them in the right order. And, you just do the best you can. You look at anything that's more similar, chances are, is more related than something that is far more different. Okay. So hopefully that's pretty simple. I don't think that's too crazy. I can't go too far because it's unique for every question. So anyway, if you have any questions, please, please, please don't be afraid to leave a comment. I don't think anyone said anything in the chat. So if you do have a question, please let me know. And the next session is going to be biochemistry. Check my YouTube channel for a date. Hopefully I'll set one. It may be tomorrow or it may not be. We'll see. So I'm going to keep an eye on this, and hopefully we can do a couple more reviews before we hit the AP exam on the 14th. So thank you for joining me, and I hope you got something out of it. So have a wonderful night.